or I can give my little 2 cents worth about what I think about future day. So when I was thinking about the future, one thing as a scientist, and I guess even as an individual, whilst thinking about the future is really, really exciting, but I think it's also important to appreciate the uncertainty and not knowing the future, because that's the fun part of science, really discovering and not knowing, not exactly always expecting something, because often the biggest discoveries are the unexpected ones. So it's always a balance between, whilst being on the path, and as Adam was saying, I totally agree, to be aware of the future and get things organised, prepared for that. We also need to allow that aspect of creativity, of the unpredictable. And I say that even especially in the context of stem cells, because what we know about stem cells today, many years ago, if someone said, this is what we can do, they probably would have burnt them at the stake and say, no, it's impossible. And so a lot of things are sort of like unexpected. And in science, you always think you have to allow that space. I say that to students, play around, to see what the experiments tell you, not just what you're trying to look at. So just again to put things into context a little bit, I'm not a historian, but my partner is, and I think if Winston Churchill was a stem cell biologist in 2014, sort of the Prime Minister in 1942, probably would have said the same quote about stem cells, where he said, now this is not the end, it's not even the beginning of the end, but it's perhaps the end of the beginning. Because in stem cells for the last 30, 40 years, we've been really trying to understand what is a stem cell. We're discovering new stem cells in the body, how to make new stem cells, what they become and so forth, which is great. But now, whilst we're still working on that, we're saying, okay, great, but now what can we do with them? It's great to have them, but how do we make this work for therapies? How do we make this work for our, future sort of understanding of science and biology? And so I think where we're at the last few years is the end of the beginning being the stem cell part. But to put things into context, especially if you think about where things are going, you have to understand where it's come from and where we are right now. It's always critical. So just to give you a little bit of background about stem cells, if I say the stem cell word, many of you would think, oh, embryonic stem cells, cord blood stem cells, bone marrow stem cells, hematopoietic, and so forth. But really how you define them is the potential of what they can become. A stem cell is a cell that self-renews, gives rise to itself, and it also has a potential to become something else. So the different categories are like totipotent, pluripotent, multipotent, unipotent. The ultra-gold stem cell is a totipotent, and there's only one type of stem cell, and that's the fertilized egg. So the fertilized egg can give rise to all cell types of the body, but also all the cell types that support the growth of the embryo. The next one down, which is the ones we work with, is pluripotent. So they're very close but different. And the difference is that pluripotent stem cells can give rise to all cell types of the body, but not all the lineages that help the support of the growth of that embryo. They can't do it on their own, okay? And then you've got multipotent stem cells like cord blood, hematopoietic, that give rise to all the cell types of that tissue, but not other tissue types. So with pluripotent stem cells, there's really 3 types. So as I was saying, we've got these stem cells in culture, and really the main characteristics is that they can give rise to all the different cell types that give rise to a whole body, all the different lineages. And the 3 main types are, firstly, is human embryonic stem cell lines. Mouse embryonic stem cells have been around for at least 30, 40 years. Human embryonic stem cells were first derived in 1998, the first line. To give you some background about that again, how do we derive them? So human ES cells come from human embryos that were created for the purpose of in vitro fertilization. It's against the law in Australia and most other countries to create a human embryo for research purposes only. So couples that are undergoing IVF treatment, they undergo IVF, and what happens in the Petri dish, you fertilize an egg and the egg keeps dividing for 3 or 4 days. After 3 or 4 days, it gives rise to what we call a blastocyst. So what's a blastocyst is a group of cells that contains an outer layer of cells and then a big cavity inside And then aggregated in one side is about 100 cells. They're called epiblast cells. Those 100 cells will keep dividing to give rise to the whole embryo. So what happens in IVF and even in animals is they freeze that blastocyst, or they take that blastocyst and implant it into the mother to give rise to an embryo. So what we do for embryonic stem cells is that we extract these 100 cells and we culture them, and we've worked out the conditions how to maintain them in culture. And these stem cells can be maintained in culture for years. So the ones we have in our lab were derived in 1998 or the year 2000. Now, obviously, there's a lot of strict ethical laws around this, and we're not going to the IVF clinic every week and collecting new embryos as people think and so forth. The only clinic at the moment in Australia that has the ethics to do this is Sydney IVF. And even though there's thousands and thousands of frozen embryos in all IVF clinics, because more and more women or couples are doing this, there's really strict laws. So to the license to create one embryonic stem cell line, perhaps they can only use 50 embryos. And these embryos were frozen for more than 5 years, and they were destined to be discarded, and then they got the couple's consent. So there's really strict laws, and I think that's a really important thing to appreciate why we're working with these cells. Now, the great thing about these cells is they have the capacity to give rise to all cell types. So from this technology, we have learnt a tremendous amount about stem cells that we couldn't have done from the animal, because there are differences between mouse and human. But obviously, it's very ethically sensitive because it was, it had the potential to become a human embryo. Now also the laws up until a few years ago, if there was anything wrong with this blastocyst, if it was carrying a gene mutation, like a lot of couples do IVF for cystic fibrosis and so forth, by law they had to discard it. In 2006 the laws changed in Australia, where now couples that find that this embryo carries a mutated disease gene, such as, for motor neuron disease, whatever, we now have the law to make embryonic stem cell lines in order to study that disease in a dish. So things are going for the better in that way. Now, as I said, we're also trying to move away from this because it is ethically sensitive. So the other way in which you can get pluripotent stem cells, which a lot of people would know about, is through somatic cell nuclear transfer, or cloning. So very briefly, the process in this is you take an egg, a female egg, you can even use animal eggs, rabbit eggs, or ideally human eggs, and you extract out the DNA. Then you take a cell from an adult, usually a skin cell or so forth, and you extract out the DNA. Now normally sperm and eggs contain half the number of chromosomes, 23 chromosomes, but all the other cells in your body have 46. Now when you extract out the DNA and you insert 46 chromosomes in the egg gets tricked, and the egg thinks it's fertilized. So it's the process of having double the number of chromosomes that we say activates the egg. And so the egg starts to divide and gives rise to a blastocyst, just as if it was fertilized. And that's how they clone animals. So then they take that blastocyst and they clone it into a pregnant female animal, and it derives a clone. But the DNA it carries came from the original adult. Now again, in Australia and all around the world, it is against the law to do reproductive cloning. We're not allowed to take a human embryo that was cloned and put it into a female. We go to prison and we pay a high fine and so forth. And so it should be like that. But what is allowed is to take this blastocyst and take the embryonic stem cells and make ES cell lines. Now this all sounds great, but the efficiency is really, really low. So far to date, only a group in America have been able to make some stem cells, but they can't propagate them. So whilst in theory this is a great technique, it still is quite inefficient. And so there's still people working on it, but we're not pursuing it ourselves as much. So the third way is a new technology which got a Nobel Prize last year for Yamanaka. And this is quite incredible, and this is what I was saying before, where you have to allow opportunities to come to you. So a group in Japan, led by Yamanaka and colleagues, what they found was that they were trying to work out what are the genes that make a pluripotent stem cell pluripotent. And you'd think it'd be 1 billion genes. But they actually narrowed it down to 4 or 5 critical genes or proteins that were involved. And the way they did this was a lot of work. They took mouse tail tissue, mouse tail cells, and using viruses, they started putting a big mix of all different genes. And they looked when this skin cell could start changing to become like a stem cell. And in the end, they narrowed it down to 4 or 5 critical genes. And so what they did, they took these mouse tail cells, or fibroblasts, using viruses at the time, they put in these genes, and they made these ES cell-like. But they're not equivalent to ES cells, so they call them iPS cells, induced pluripotent stem cells. And from these stem cells, they could make a blastocyst and create a whole new mouse. So essentially, they created a whole new mouse from the mouse tail tissue. Now, already a year later, they were starting to do that with human cells, and same with American groups. The whole stem cell field, all of us stem cell biologists, I was at the conference when you first presented this, we got on board because we thought, oh my God, this is unbelievable. Because you can imagine using this technology, we can take your skin right now, which we do in the lab actually, and we introduce these 4 or 5 different transcription factors, we make them into stem cells, and now from these stem cells, we make them into any cell type of the body. And the idea behind this is that we can take this, make it into a liver cell, a nerve cell, whatever, and put it back into your own body, and it won't get rejected because it was originally your DNA. Right? The other cool thing about this technology is we've come a long way knowing how to genetically manipulate cells. So if you're carrying a mutation, we can work out how to fix that mutation. We can insert different genes. We can make your nose green if we want to. We can do all the things we're doing with mice into the human cells and put them in, right? But saying all this, there is many groups. This was all very cool and exciting, and it's coming a long way. But one thing you have to appreciate, every arrow represents massive amounts of labs just working on this one arrow aspect and many years of work. So there's groups that are just dedicated to reprogramming. Groups like myself that's trying to dedicate and saying, okay, we've got a stem cell, how do we make it to a cell type of the nervous system that's relevant to that disease? A lot of years of work, genetic manipulation, transplantation. But where this work is really heading is saying now we have a way of getting human disease cells to understand basic disease processes for drug discoveries and even understand how things develop. In my case, I'm trying to use these stem cells to understand how do we get a whole brain from a stem cell? How does that happen? So, to talk just brief overview about my work and the work we do. So it's a big cut off, you can't see. But that's my job, essentially. We have these pluripotent stem cells. We work with both human and iPS, skin-derived ones, they're all human. And we're saying, how do we make them into the relevant cell types of the nervous system? The nervous system has 1 billion different types of neurons in the brain and spinal cord. How do we drive them down? It's a double-edged sword. It's great that they've got the potential to become everything, but it's also a big, big problem because you have to try and work out how to do that now, right? And it's relevant, so we work on dopaminergic neurons for Parkinson's disease, oligodendrocytes for multiple sclerosis, and then another lineage we work on is, that I'll talk about is the peripheral nervous system, all the nerves outside your body, for things like hearing loss and Friedreich's ataxia and so forth. How do we do this? Well, basically, my background, and this is what we have to do, is we have to come back to the embryo. So we go to all the years and years of studying how a mouse develops, how a frog develops, how a chick develops, and we understand how those brain processes occur. And we look at the brain and spinal cord, and we look at all the different factors, all the different genes, and we try and mimic that in a dish. We add the different genes and at certain time points and sway it to different parts of the brain and different parts of the spinal cord. I'm not going to go into that because it's all scientific, but that's basically, in a nutshell, what we do. And then in the lab, we can then mimic that same process. So we've got our stem cell colonies. Here's a colony of stem cells there. So there's about 50,000 cells. These are derived from the skin. And there's a human ES cell colony, another colony of cells, about 50,000 cells, but they're ES cell derived from an embryo, from a cell line from 1998. Then we add certain factors to switch them to go into sort of neural differentiation, neural progenitors. And then we can take these stem cells and then they're restricted. They're not going to become muscle. They're not going to become liver. They're going to become cell types of the nervous system. Then we aggregate those cells. So they're still stem cells, but they're stem cells of the nervous system. We culture them in suspension, and then we add other factors to drive them to become a neuron, to become a glial cell, to become a motor neuron, dopaminergic neuron, and so forth. Now, just to put into context, one of the diseases I mentioned we work in, why we do this, is like a disease called Friedreich's ataxia. So it's a very rare disease, it's about 1 in 30,000. So the word ataxia means loss of balance, right? And it's a genetically inherited disease. So basically the symptoms of this disease is that usually the symptoms occur around childhood, and these kids start losing balance. They can't walk properly, they're fumbly, and then over a few years they have to be in a wheelchair. And they also have severe heart problem, and their life generally is about 20 years or so, and there is different degrees of severity. Now, not to go too much into it, but why they get this disease is because they carry mutations in this gene called frataxin. Now, in the cell, you've got different organelles that do different functions in a cell, and one of them is the mitochondria, and that's like the energy pump of the cell. And this protein, frataxin, has a role in maintaining iron levels in mitochondria. So basically, to cut a long story short, these patients have mutations in this gene, and they have very low levels of this frataxin protein. So they get iron toxicity in these cells, and this iron toxicity causes cell death. And even though frataxin is in every cell of your body, there's some cells that are much more sensitive to these low levels, like the heart cells and some cells of the nervous system. Now, the reason we work on this, apart from, well, there's many reasons, but one thing is, even though it's rare, it's actually low-hanging fruit, because unlike many other diseases like multiple sclerosis or motor neuron disease and so forth, where we don't know the causes or the genes involved, here we do know the gene and we know essentially how to cure it. All these patients need, a bit like diabetic needs insulin, these patients just need extra frataxin protein, not even 100%. They need just like up to 30, 40% to cure it, or ideally to cure fix the mutations in the gene. Or they can be treated with antioxidants. So because of this, a lot of pharmaceutical companies, especially the parents with these sick kids, know that there's a chance, there's a massive push, a lot of money to find a drug for this disease, where they can find compounds to increase the frataxin levels and alter into the DNA. So over the last few years, they've looked at different compounds, antioxidants or HDAC inhibitors that interact with the chromatin in that gene region and so forth, or delivering frataxin itself. But what's been holding up the progress of this is having an appropriate model system. So they've made mouse models of this disease, but they don't tend to show the symptoms. So the closest thing they've got is blood cells and platelets. But again, it's not relevant to the disease. So here's a good candidate where stem cells can come in to sort of find this drug cure, basically. And so we got funding for this, and that's what we did. We got skin samples from these Friedreich's ataxia patients, and we reprogrammed them to become stem cells, iPS cells. And now what our job is we've got the stem cells, and now we can't just work with the stem cells. The whole point of this exercise is to make them into the relevant cell types that's relevant to that disease. And that's basically what we're doing. We're making them into different types of cerebellar neurons, and one type which is sensory neurons. So again, in this disease, the type of cells that die are we call dorsal ganglia sensory neurons, and these are cells that are found just outside the spinal cord. And there's different types of sensory neurons, ones that help dictate pain, but the other types that help dictate feeling and pressure. So the reason you're sitting in your chair right now is because you can feel the chair underneath you. You can feel the floor underneath you. It's all subconsciously, but it's there. Can you imagine walking and not feeling the ground under you? You wouldn't be able to walk. So the nervous system is unbelievable. If I shift my weight gently on one side, the body knows to compensate so I don't fall over. It's unbelievable, the nervous system. So if you lose these type of neurons, you will lose a lot of balance because you're not getting any feedback mechanism. You put your foot down, but there's no feedback to say, you've hit the ground now, lift up the other leg and walk. That's what's going on in these patients, right? So, how do we make sensory neurons? So, I'm not going to go into it, but basically we go back to understanding how these cells develop in the embryo. And we go from stem cells, we have different factors, we go to neurolectoderm, then to neural crest progenitor, peripheral neurons. And peripheral neurons is every cell neuron in the body that's outside the spinal cord. So that includes neurons that go to the heart, the gut, the dorsal ganglia, your mouth, everything. So there's a lot going on that you have to still try and sway them into that lineage. And we're getting there though, we're doing it. We've worked on techniques recently where we can take these stem cells and we can make them into neurons. Use a group of neural progenitors, and you can see they're extending out their axons here, their neurons, and they're expressing markers that are relevant to these dorsal root ganglia. So we're quite excited because we're onto it. And from there we can take these neurons and we can start looking at their functional properties and seeing what's wrong with these cells so that when we just throw on the drug, we can see a reverse, okay? But what's been cool about working on this, that's one thing, differentiation, is that it's also relevant to other cell types. One example is the auditory system. So I collaborate with Bryony Nagam and her group at the Ioneer Hospital who works on the cochlear implant. Now with the cochlear implant, there's a lot of reasons people get hearing loss, and it's basically loss of hair cells, but also loss of these auditory neurons, spiral ganglion neurons. Now, when you get the cochlear implant, you still get stimulus bypassing the need of these auditory neurons, and it goes to the main auditory nerve. But over time, these auditory neurons still die because they're not getting any factors delivered to them from the hair cells. So what she's trying to look at is complement the cochlear implant with stem cells to replace these nerves. And these auditory neurons are very, very similar to the sensory neurons that I was just talking about involved in feeling. Very similar properties. So she's been taking our system and our stem cells and she's been putting them together with regions of the skin, the epithelial tissue from rats. And she can see that these neurons are innervating into the skin and making connections with these hair cells. So that's pretty cool, and that work's ongoing now. She's doing some in vivo work. The project that I collaborate with is Don Newgreen at the Murdoch Institute at the Children's Hospital. And he works a lot with Hirschsprung's disease. So Hirschsprung's disease is another genetic disorder where, well, they think it's genetic, they don't know the causes, but basically babies are born and for some reason they have a lack of neurons, enteric neurons in the nervous system to the rectal regions of the gut. So these babies are born and they have severe constipation that can kill them. So they have to do immediate surgery where they remove that rectal part and place other parts of the colon, the large intestine, into the rectum. And it's awful because they can still, even though it saves their life, for the rest of their life they're having many other complications. So what Don's working on is, can we replace these neurons at that early stages? And we can with stem cells. And we're lucky because these enteric neurons, we use a very similar system that we make to make these sensory neurons. We use the same system to make these gut neurons because they're peripheral. And so we did this. We used this system and we made them into early, we say neural crest progenitors, and we put them, these are the green cells here, and we put them together with mouse gut tissue, embryonic mouse gut tissue. And we found that these cells migrated into the gut and they start differentiating into nerves of the enteric. So we're really excited with this work, and now he's actually going into human tissue that he gets from surgeries to see if it works there. And there could be a potential for replacing these cells. All right, so as you can see, this aspect of differentiation is really important for drug discovery and modelling. And because of this, many groups around the world, not just us, have been making iPS cells from all different diseases, all of them. And this is just limited to the nervous system, but even diabetes and so forth. And a lot of these, you can't see here, there are even complex ones, like even schizophrenia. Very, very complex. And I myself have to put up my hand up because we as well are starting to work on autism spectrum disorders, and say, how on earth can you take these stem cells to model ASD behaviours? Highly complex, right? But that's what we're doing. We've got some fibroblasts from skin cells, At the moment, we're going to get them from Australia, but at the moment there's a big bank in the US that's providing them. And in the lab, we're starting the reprogramming them to make them into stem cells. And then our next job is to make them into cortical neurons, the neurons that are relevant in autism. And I have a PhD student that's working on this. So again, the brain is very, very complex. There's all the different regions of the brain, but even a slice through the brain, we've got at least 6 different layers, 6 different main cell types across the brain if you look on a vertical axis. So using our stem cells, we're looking at, okay, we're getting cortical neurons, but what type of cortical neurons do we get? And once we get this, then we can look at their function and interaction in autism versus control and start to model behaviour from a synaptic point of view, because there's a lot of theories out there saying, is autism because there's too much synaptogenesis? Is it because it's the wrong cell types? Is it because there's too many inhibitory neurons? We can somehow start tackling in there, because obviously we can't get the brain and open it up and look inside. You know, we can do some imaging analysis, but here we can do it in a dish. And the technologies, like recently, last year moved, as Adam was saying, to the Centre for Neural Engineering. And I'm really excited about this because I need the engineers' help. I'm a biologist, I'm not an engineer, you know. And so we have to work together for this, for the modelling. And so people, when you look at function of cells, people traditionally have been using a technique called patch clamping, where you take this electrode and you point to the cell, and you zap it, and then you can measure the different action potentials. But now we're getting into more sophisticated techniques. They're building it. We're going into nanowire technology. So what we've got engineers doing is they're building these vertical nanowires with special material. Like, don't ask me what it is, they know. And they can poke into the cell, and they can not only measure the electrical stimulus, but they can measure the different chemical properties in the cells to look at changes. Also, what we're doing from there is we're working with microelectrode array technologies, which is a new technology. So what we've got is we've got these arrays of 60 electrodes, and we culture our stem cells and neurons on there, which we have. And then what you do is you zap this electrode and you measure activity here. Or you can do spontaneous firing. So here we're starting to measure connectivity, a network where neurons are really talking to each other. It's not just an individual neuron. And this is actually really, really great, because where we're going with this work now is we're building these stem cells in 3D matrices. So we're building the different cortical neurons, and we're looking at different polymers to grow them in. At the moment we're using silk, because silk is a natural substance that they're already using in transplantation, and it naturally dissolves. So we're culturing those different stem cells in silk layers, and then we'll be able to zap them and say, okay, let's stimulate up here and record down there, or let's see the electrical activity that they're doing. So we're basically modelling the brain and building the brain with the stem cells, if you want to put it that way. And then we can look at differences in behaviour. So that's basically where the work is going. But many of you may be thinking, well, what about transplantation in stem cells in general? People doing this, because I've been talking about modelling. And whilst we're working on that, for iPS cells, we're not quite there yet. Because whilst they're very, very similar, they're not 100% the same. And there are some issues that we have to find out, because they're basically the way we make iPS cells is changing the whole DNA. It's never 100%, and you get a lot of variability. But that hasn't held up the field, because in America, from when human ES cells were first arrived in 1998, already by 2006, less than 10 years, we got iPS technology where you can now make stem cells from skin. And in less than 10 years, they're already, in California especially, there's billions of dollars going towards stem cell therapies, and they're pushing them for clinical trials. So interestingly, the first clinical trial for stem cells was in spinal cord injury, where they found they could make a cell type that could help promote regeneration. But unfortunately, that trial was soon stopped because they claimed that spinal cord injury is not, doesn't occur frequently enough to justify the amount of money that they're putting in, right? And we were all disappointed, because it would have been really good proof of principle just to see that stem cells can be transplanted in. But where they're going now is that they're putting a lot of funding into stem cells for treating macular dystrophy, because using human ES cells, they can't use the IPS, they can make retinal pigmented epithelial cells. So these are cells that just are on the surface of the layer of the eye, so you don't have to worry about going into the blood-brain barrier. It's much more easier. And it is looking quite promising, that trial. And very recently, they've just got a funding to try a clinical trial for using stem cells, human stem cells, to make heart cells, cardiomyocytes, for severe heart disease, to see if they increase. So it's on its way. They're using human embryonic stem cells for now because they know it's safer, but eventually they will go to IPS. So this is where the whole field is doing, and basically that leaves me with the question, what can we do with stem cells? But I'm going to end on a provocative note and say, well, I think we're soon going to change the question and say not what can we do with stem cells, but actually say, do we actually need stem cells? Because where people are now going with this whole concept of reprogramming, where you can take a skin cell and make it a pluripotent stem cell, has changed the way we think about cells. Things are don't always stay the same, and what people are doing is now they're taking skin cells and they're bypassing the need for the stem cell and they're making it directly into a nerve cell. And they're already doing this to make dopaminergic neurons and a pancreatic cell. So this avoids this whole worry about how do you go from a stem cell into a particular right type of cell. We're still not there, but there's a lot of people doing that. So can you imagine, we take your blood and make it into a nerve. We take your muscle and make it into a liver cell. We take anything and make it into whatever. A fat cell, everyone would love that, you know, make it into hair cells or whatever. So that is where this technology is going, because this whole concept of reprogramming, transdifferentiation, shows that things can be changed. Things can be, it's all about manipulating the DNA, exposing the right genes. So it changes the way you think about things, even ethically. Well, what is a cell? What is a person? And so forth. So that's why I was saying, I'll come back to this quote. It is the end of the beginning. We've worked many, many years understanding stem cells, but now we realise it's not just about stem cells, it's actually about reprogramming. It's actually about DNA. It's actually about cell function. We're not defining things as strictly anymore. And we're thinking of new ways to go further with this work. And I end on this slide. What's really nice, stem cells is very ethically sensitive. You know, we had to go deep. We had to go there. Like everything, it's a bit like, you know, science is like that. It's a very sensitive issue. But what's really nice is that you can give back. You can give back. It's through stem cells that we can now work on therapies to create organs, to understand nature, to understand who we are and what we come from, to manipulate. And I think you need to go there, always, you know, in the right, proper way. And so I'll leave it with that, and I always acknowledge the people. There's always lots of people involved in work and funding and so forth. So it's not me alone, it's always a massive team effort of different groups with different expertise that work together on this. So I'm open to questions. Thank you.
So I just want to get your talk clear in my head. So is the idea that, so differentiation means roughly programming a cell, right? Yes,
yes. Yeah, that's a good way to put it. And so if you go backwards, you say de-differentiate. It's programming a cell in one direction though. It means that it's going from a stem cell to a more mature cell type. But if you go backwards, it's de-differentiation. But nevertheless, it's a path. And programming is a path, really. Yeah. Yes.
Yeah. If you don't, so there's this. How do we get more IPS cells?
In terms of what do you mean by how do we get more IPS cells? Because we can derive lots of IPS cells from, you know, and people are working on optimizing how to get better IPS cells. Because it's all about how you really manipulate the DNA, and
I think
so.
Sorry, I think I misunderstood your diagram. So the idea is that we take, say, a little bit of skin, say.
Yes.
And then we get a whole bunch of IPS cells from that.
Yes.
And these these stem cell-like things form the raw material for differentiation.
Exactly, because they're the cells, they're very similar to embryonic stem cells that give rise to, from there we have a source to give rise to all the different cell types that's relevant to whatever you want, really. Or, you know, that's the theory, the model. Yes.
What is the construction management process of an organ and also the entire process?
Yeah, that's an excellent question. So, sort of words that I like to use with that is intrinsic and extrinsic. So sometimes cells are programmed, right? They have the genes to dictate what they're going to become, but they're also influenced by the microenvironment that they're in. So it's a play of both things going on. And the other word we often say is commitment. So often cells are uncommitted. They can become anything. Then you put them in the appropriate environment with a signal that says, all right, now it's like being in a country. I'm going to be an Australian, born in Australia, you know, and you learn certain things, but they're not necessarily committed to that lineage. Then they get other signals, intrinsically or extrinsically, and then they become committed. So no matter what environment you put them in, that's it. They're a liver cell. You put them in the brain, they're still going to be a liver cell and they'll just die. So there's always these things of plasticity to a certain point. And that's how an organ develops. It's sort of, firstly, it's open to all possibilities, and then it's exposed to a certain environment because of the signals, the way the organ is, and then it starts differentiating, gives rise to different cell types, and then it gets to a point where it becomes committed, and that's it, it's on its path. The other thing is there's a lot of balance, stoichiatric sort of thing. So, like the nervous system, for every nerve it makes a supporting cell. And it also, even in stem cells, they replicate and replicate and replicate, but they also give off a more differentiated cell type. But then eventually that runs out of steam. So it's a little bit like a tree, you could say, you know, a trunk, and then it decides it's going to make branches, and then it stops and decides it's going to make leaves. And I think it's a balance. Okay, well, we've got enough of these cells. Let's start making these other cells. And then you're getting into the DNA where you're looking at telomeres. There's different intrinsic things in the DNA itself that has a certain life. It has a certain path. And that's the whole craziness about reprogramming, because you change that properties of the DNA. It's like a tape, you wind it backwards and give it the same properties as a stem cell.
In computer programming, you have a whole suite of algorithms waiting for you to use, but you don't use them all at once. You use parts of them at once. It's the same process with the DNA too.
Exactly, exactly. And then I'm sure that if you, well, I don't know, I'm not a mathematician or computer scientist, but if you use certain algorithms, does that lock you or inhibit you from using others? You know, because in the stem cell it does that a little bit. Like, in progenitors, if it decides to become something, it's locked into doing something else. So you have to physically go in and deprogram it to give it all the, you know, or doing this makes it do that and that and that.
So it's a recruitment process.
Yes, exactly. Yes. I'm sorry.
Well, I'm not in the field, there are a lot of claims about the computer.
Yeah.
I'm a subscriber to science, stumbled across this article. That's embarrassing.
No. Yes.
It's George Church at Harvard and Charles Hirschsprung at Duke. You're looking at CRISPR-Cas9s,
Okay.
bypass pluripotent cells,
Yes.
bypass RPS, and go straight into genome editing.
Exactly.
What's the prospect for that?
Oh, it's great. That was that second last slide that I was showing with the nerves. That's what they're doing. So they're using CRISPRs to target DNA, certain genes, to switch them off or not, on or off, and then you don't need to go through a stem cell.
Yes.
But we need to appreciate that the only way we know how to do that was to understand. It's like, you know, everything else, and we still don't know if we've got all the players right. Like a lot of people try to make dopaminergic neurons, and just because these cells do this, that, they go, oh, it's a dopaminergic neuron. But really, truly, it has to show all the properties to be truly functional. But we're getting there. That's where we're getting, because it's too long to take a skin and go all the way through. We're doing it because that's what we can do now. But ultimately, we will bypass everything and take your skin and make it a pancreatic cell, or, you know, yeah, that's
you
it.
see this is the possible cure for preventing troublesome cancer?
Yeah, cancer's an interesting one because it's very, very similar to stem cell properties. I think we can learn a lot about cancer. We're only now starting to collaborate with the cancer group that work on pediatric tumours, and we're trying to model these cancers with our stem cells. And I think we'll learn a lot in terms of understanding how to treat the cancers. Because if we can mimic and create a cancer from the stem cells, then we've got the source of the stem cell cancer, that we can work out the different therapies, especially radiation and so forth, to target that cell type.
Who's working on it right now?
Yeah, I've started a collaboration with the group at the Department of Pathology. Just on the side with someone who's, the cancer person came to me, and there's a group in WA that works on paediatric tumours, and I've been giving our stem cells to him so that he can mimic that paediatric tumour. I can't be everything, you know, and so it's always collaborative, and I say, okay, I can contribute to the stem cell side of it. They work on the cancer side, a bit like the Hirschsprungs and everything else. You still put your fingers in many pies and say, yeah, I can talk about this, and they talk about that, and you come together, they can share a student, and they just drive the work. Yeah.
Can I ask a little bit about engineering? So you have clone your DNA, so you have genetic disease, and pull it out, and you isolate that, and fix that gene, and then you put that gene into some other cells.
Yes.
What I'm going to put back into your body, how does it put place in the whole DNA?
Yeah, it's an excellent question. So there's different ways people are doing it. So people with gene therapy, they go in directly with the virus that targets your cell. That would be one way to do it. Or adenoviruses. The tricky part about going into your actual DNA is that you don't want to modify your DNA. Like viruses, you don't want to interrupt genes. So one way around it is you can have DNA just floating around, sort of like viruses do, and they can create the protein, because we can engineer that DNA to make frataxin or insulin and just sit there and spit out. And then there's a lot known because we can modulate what we call the promoters and different things that regulate the expression of that gene. Because, for example, you don't want a virus in there pumping out all this insulin. You want it to respond to, you have some sugar, let's make some insulin. So there's a lot of different modifications. If you modify a cell, now they're very, very clever. There's different ways in which you can insert genes in, and then you can come in with other things and take out any parts of the tools that you use to put it in. It's like you open up the DNA or you change it, and then you can excise out the parts that you put in. They're unbelievable, and we know this because of all the studies with, you know, deep marine sort of organisms and other like organisms that do that naturally, and flies and Drosophila viruses that do that naturally, rabies virus, all that, HIV. So we use that as tools to play around with. There's a lot that can be done. Yeah, sorry, I've got one at the back or something.
During the 30s and 40s, when physicists started getting close to understanding how atoms work,
yes,
many of them who started off as pure scientists started thinking about the system they were operating in and what created it, starting about the mind of God and stuff like that.
Yeah.
It sounds to me that how are biologists reacting?
Yeah, it's a great question. It's a great question when I give lectures to students. I think when you question life, you question what makes you you, if we can make stem cells from skin and make a human embryo or human, you know, what is all of this? Who are we? We're a bunch of cells put together. The scientist in me says yes, you know, but the other scientist in part of me says, actually, it's how things are put together that matters. It's the connectivity. It's not just the cells themselves. It's how they function and work together that makes you you. And I think you can go even further, and I say life never started or died. You're truly, if you want to become philosophical, you're from your parents' DNA. It's an ongoing process, and it's all a merge, everything, really. but it's how things are put together that makes you an individual, if you want to see it like that. And so it's really fascinating. And I think as a scientist, you become far more respectful about life, far more respectful about animals. You work a lot with them, far more respectful, because you're just in awe of how things happen. And you think, yeah, it's not just me, my little world. It's actually, nature is unbelievable. And where it's going, you have to take a step back and think we're not the supreme race. I think there's so many organisms that do it so much better. There's a little, I can't remember, a little thing in Japan that they were studying where it wasn't a worm, I forgot the name, and they chopped it up into 100 pieces and make 100 new worms. It got too fat and it just divided, and it was far better regenerative medicines than us, you know. So you appreciate these things in nature, really. Yeah. Sorry, yeah?
Just on the practical, you're talking about liver transplanting in organs. So how long does that take? Okay, I've got liver failure,
Yeah.
or I've got kidney failure, whatever the part is. Okay, here's some skin. How long do I need to wait?
Yeah, it's a great question again. That's why people are not thinking IPS will become personalised, because the actual process is very practical, takes at least 2 months or whatever, but also everything has to be done animal-free in GM facilities, and it's a very, very expensive process, and it's not practical. So the way they're thinking about it, like in Japan, what they're setting it up, is to make a big bank. So like, for example, bone marrow cells, is one of the limitations in having transplantations to find the perfect match. So if we can have massive banks of IPS cells, maybe you can use that to find an ideal match to then transplant into you. If you had enough money, it would be personalised, but I think it's really becoming not practical. It may do, but I think people are really not thinking like that. I think there was a question there, sorry.
Sorry, yeah. Just wondering if the telomere and the cell, lifespan.
Yes.
So if you transfer a skin cell to another
Yeah.
cell, that
Goes back to its original state.
Okay, so
It's quite incredible.
You start young.
Yeah, it gives you more life in theory. Even the mitochondria, it's unbelievable. It goes back to its original state. It's unbelievable. Yeah.
So the telomere is evaporating.
Yeah.
Look forward to the latest planet. Printed lake.
Yeah, that's it.
You say, go there and live there, go there.
Yeah, it's unbelievable. It's not my filbert telomeres, but they go back to their original. Yeah, sorry, I had it.
Related question, I think. Years ago, I was told that the stem cells, when they divide, are subject to limitation called the Hayflick limit.
Right.
What is the status of that idea today?
The Hayflick limit.
Yes.
Yeah.
Fixed number
Oh.
of divisions.
Divisions, yes. So it's the same. Yeah. Things are also different because we can control things in a culture dish. So whilst we go back to stem cells, we try and give them the conditions to arrest them in an earlier state to make them divide longer. So we can manipulate them, but if you just let them be, like our stem cells that we grow, every week we have to passage them because they do tend to differentiate on their own. They don't just stay there and now they're not arrested, they're proliferating, and as they're proliferating, they're already giving rise to different cells. Sometimes you look down the dish and you can see, some have gone neural and some have gone extra. So you cut out the regions that are still ES cells. They tend to do that. Then if we want to keep them into neural for longer, we know what are the conditions to keep them for a few more passages, and even that's got a limited life. So we can only do so much, but we can trick the cells by changing the microenvironment to make them proliferate longer, and then, but eventually they'll run out and do their own thing. So it's really like it's like animals, you're trying to herd them, you're trying to put the brakes on, or you're trying to, but they'll still do their own thing, and you're trying the best. It's like that. So the cells do differentiate spontaneously. That's a good thing, otherwise it'd be cancerous. We want them to differentiate. Yeah.
Does this kind of process ever stop in its tracks stop there because of any particular reason?
Oh yeah, we get like sometimes the cells, you know, come in and something's happened, and again, it's stem cells. It's very, we don't have a perfect experiment, perfect lab. You know, it's like you go in and some weeks the cells don't do as well, you know, and you don't know you've done everything right, and then the week after they recover, you know. Things, you're still ultimately working with nature. You really are.
I was really asking about the body.
Oh, what do you mean, like the body? Like if it stops in its tracks, sort of?
Any of these processes so that it causes problems?
Yeah, like I think like certain organs and things, again, like depending on the microenvironment, I think they will respond to that. And it can sort of do different things. The body does strange things. If that's what you're asking, I'm not sure, like in...
Well, if, for instance, you ingest the wrong thing,
yes.
And it interferes with the process.
Right, yes, for sure. Like if you look at even children, developmental defects, you know, if they were malnutrition, the body tries to compensate always, but it can only do so much. There's a lot of redundancy in the body, and it's quite incredible how it can compensate, but ultimately, if it's not fixed, disease will happen, things will happen, for sure. Yeah, sorry?
Is anybody working on the fields, the electromagnetic fields with this?
Yeah, so I'm interested in that. One of the groups in our Centre for Neural Engineering is a physicist, and he works on the electromagnetic field in a single neuron, and how a single neuron changes its magnetic field. And I've been talking, and he uses, you know, neurons from frog legs and things, and I'm saying it'd be nice to do that in our stem cells in the neurons to look at. And there have been, you know, that would absolutely influence the properties of a cell and a neuron. Not that I've studied that, but it'd be really fascinating to look at that, because I'm sure it's...
Is that Colin Harms?
No, it's Lloyd Hollenberg. Yeah.
In fact, Lloyd spoke at the Science Technology and Future
Oh,
conference
right, there
just
you
recently,
go.
and he was absolutely awesome, so look him up.
Yeah, he's excellent. The work he does is unbelievable. He's working with diamonds and something. Yeah. Patrick?
Yeah. So,
Can you speak your questions up a little bit so the microphone catches it?
sure thing. Is this good?
Yeah.
Great. So, sorry to, I just want to get this, I just want to have a few pictures in my head. So, I imagine cell differentiation works sort of like this. We surround a stem cell with lots of nerve cells, say.
Yeah.
Is that stem cell much more likely to differentiate into it?
That's a very good question. Some cells secrete factors that promote the neighbouring cell. Well, all cells secrete factors. They all influence the microenvironment. We think yes and no. We think at certain early stages, if they're put in there very close to very early nerve cells, they will, because these nerve cells by default start secreting to tell the neighbouring you become a nerve and so forth. But then it gets to a certain point where that influence no longer happens. And yeah, so really in a culture dish, we create that microenvironment to make them become something. And so in our conditions, yeah, it depends. What I keep saying to students, it depends not just the what, but the when is critical. When is, there's certain cells that respond to things at certain stages, and then they no longer respond, or they do, and so forth. So it's not just a fixed thing.
So differentiation depends very much on the chemical environment.
Yeah, and even cell death, like if we've got too many cells dying in a dish, even the healthy ones will end up dying because they're releasing the factors to say, so that's why we have to change the media, because they're saying death, war, you know, kill, die, you know, and so there's a lot of things that, yeah, the microenvironment does play a very big role. Yeah.
Right?
Okay. Yes.
I am doing that in just a moment. I'm going to wrap it up, but thanks for all your questions and thanks very much, Dr. Mirella Dottori.